Hi, good afternoon, Honorable Show Laureates, distinguished scientists, forum participants, ladies and gentlemen. Welcome to the trending topic session of the Hong Kong Laureate Forum. My name is Laura Christensen, your MC for the session, and I'm so glad to see you all again in the same room. On behalf of HKLF, a warm welcome to all of you who are joining us here in person, and a wave to those of you that are joining us online through the live webinar. Welcome. This morning, we had a wonderful plenary session and masterclasses to discuss a whole wide range of scientific subjects, thought-provoking exchanges. This afternoon, we will be drawing our masterminds from our laureates and renowned experts to discuss a trending topic of the year, new technology and AI. And we have divided the trending topic into two parts. First of all, our speakers will give a presentation and sharing on the title of Impact of New Technology on Scientific Development. And for our second part, our speakers will share their views on AI and the teaching of science. I believe you all feel the same as I do, that these are indeed hot topics for discussions. In this trending topic part one, we're very honored to have three laureates to share with us their research as well as their views on the impact of new technology on scientific development. And I would like you to join hands to welcome them. They include Professor Simon White. Thank you, Professor, for being with us today. Professor Peter Hegeman, welcome. And Professor Victoria Caspi, welcome. After the presentations, we have the honor of having our distinguished guest, Professor Wang Yang, to host a session. Welcome, Professor Wang Yang, to deepen the discussion that we've heard from the three speakers. And just a note that after the three speaker sessions, you will have the chance to ask questions to the panelists. So I highly encourage you, when they are presenting, drop notes, make remarks, and mark any questions that you would like to pose after the session. So either you could raise your hands or utilize the app in the HKLF app Q&A function to pose questions to our panelists. Now, without further ado, I would like to invite on stage Professor Simon White, 2017 Scholl Laureate in Astronomy and Emeritus Director of Max Planck Institute of Astrophysics, to share with us on characterizing and observing the smallest dark matter objects. Professor White, please.
Well, so let me begin by saying what the relation of my talk is to trending technology, because there's some tension here between topics and the presentation of the session. So I'm a scientist who works with computers to try and model the evolution of astronomical systems through direct simulation of their behavior. And while we heard earlier from Professor Genzel about how instruments have made enormous strides in the last year and how the new sensitivities, the new capabilities allow us to see new things, I always, as a computer user, like to start by noting that whereas telescopes over my career have increased in collecting area by a factor of 100, increased in the sensitivity of the instruments by a factor of 100, have increased in the angular resolution they can achieve by factors of 10 or more, depending on where you look. These are all huge factors which multiply together to increase the sensitivity. But over the same time period of my career, computers have increased in speed by 1 billion, 10 to the 9. The memory capabilities of the numbers they can store have increased by 10 to the 10. And the total storage of the data sets we work with now have also gone up by 1 billion. So the computer resolution, compared to other aspects of many sciences, in terms of the number of orders of magnitude that have changed, has been enormous. And this has changed the way, and is still changing the way many of us do science, because the kind of modeling that you can do on computers is just unrecognizable from what was possible even 10 or 15 years ago. And over a career as long as mine, it's mind-boggling. So let me start by showing you a picture which those of you who are at the morning session on astronomy will have seen already. And this, I think, has a good case for being considered the most dramatic picture in physical science. Why is this? It's because this is a picture of the edge of the entire visible universe. Right? This is a direct image of what essentially the clouds which limit the part of the universe that we can see, the far side of the universe. They're so far away that in fact the time we're seeing it, the universe was only 400,000 years old, instead of being today 13 billion years. So here in this image, here taken with the Planck satellite at microwave wavelengths, you're seeing the universe, the entire edge of the universe, as it was when it was only 400,000 years old. And at that time, there were no structures at all. It was almost uniform. And the only structure you're seeing here are just weak sound waves propagating through the nearly uniform medium. So you're seeing the whole universe before any structures of the kind we're familiar with formed at all. So all the structures we're familiar with, stars, galaxies, planets, people, all formed later, after this time, from what you're seeing here. So how did the structures we know form? Well, here's a movie which starts with a near uniform distribution, and inside the computer, it's following the distribution of matter in a representative region of space. If I click to start it, what you'll see as time progresses, the structure slowly grows in contrast. And at some point, you'll see individual objects forming. Well, this is a large region of space, so the smallest objects you can really see correspond to at least a massive galaxy or to a cluster of galaxies. And you can see the matter is evacuating from the regions which are now red into the regions, this filamentary structure, which reminds some people of neural nets, but probably not the neurologists. And then the matter flows along these filaments into these nodes. And so this thing at the middle would correspond to a rich cluster of galaxies today. So we start from these sound waves at the early time, the structure gradually amplifies, falls together to make dense objects. So if we look at a particular object forming, here's another movie. This will correspond to the formation of an object about the mass of our own Milky Way. And here we're only seeing the dark matter component, because at the time this was done, it was too complicated to try and calculate what happens with the stuff we can actually see. So this is the evolution of the stuff which dominates the amount of material, but which we don't see directly. And so if I stop now, briefly, you can see this is what redshift 13. This is a time when the universe was 14 times smaller than it is today. So only a few 100 million years old. And the biggest lump here, these things that are yellow, these have about 2.5 billion solar masses. That's what this number on the bottom is here. So our galaxy today is about 1,000 times more massive than this. So the total amount of mass in the lumps is only 1,000th of what there's going to be at the end. But now watch what happens as this object builds up. You see all these things gradually fall together and become larger and larger. We'll just let it run for a bit, and you can see now that there are only a few very large objects, and about sometime now, one object comes to dominate all the others. And this is the object which would be our own Milky Way, and at the time we're looking at now, it's got about 1/10 of its final mass. And you see from this stage onwards, this is the last, at this stage the universe is 2 or 3 billion years old. The evolution is lots of small things falling into the big thing. And remember, this is only the dark matter. I haven't yet calculated in this calculation what's happening to things we would actually be able to see. And from now on, it essentially looks more or less like this. Let me just skip to the end, in the interest of time. You can see here we are at the end. There's a central part, our own sun, and all the stars would just be in this middle blob, and this is the dark matter distribution expected to be around our Milky Way, filling space. Now, if you look at the structure of this object, you can see it was high density in the middle, and the density fell off towards the outside. If you just take spherical shells and average the density from the center outwards, you get what's called a density profile. And if you plot it logarithmically, so this is the log of the density vertically and the log of distance from the center horizontally, you can see you have a curve which varies gently with radius. It's shallower in the center and steeper on the outside. It turns out that it's quite well fit by this formula, which some colleagues and I found from fitting such simulations almost 30 years ago now. And if you look at this, it goes like a slope parallel of r to the minus 1 in the center, r to the minus 3 on the outside. Okay, that's one case. It turns out, if you look at things of different masses, they all look the same. So here's a simulation where here's a large part of the universe, up at the top, which we've simulated. This is 150 megaparsecs, that's 450 million light years. So it's a large patch of the universe. But now there's a little circle up here. If we take this region, zoom in on it, and simulate it at higher resolution, we get this. And now this is 45 million light years, 15 megaparsecs. You take a small region here, zoom in, simulate it at high resolution, we get this. And you keep taking regions and zooming in, you get to higher and higher resolution in smaller and smaller parts of the universe, and you can see smaller and smaller objects. So by the time you finish this procedure, you've actually spanned a scale here of about 4 million in linear scale, and a mass scale of 20 orders of magnitude. So these objects down here are 20 orders of magnitude less massive than the ones you see up here. And yet when you plot up their structure, it turns out they all look the same. This is plotting not quite the same profile as I showed you before, but it's still plotting density versus radius, and this is actually the slope of the density profile against the radius. And when you take away the overall scale of the system, you can see all these curves lie on top of each other. So you get apparently the same structure of the objects over all the way from the smallest ones, which are Earth mass, through things the size of the Milky Way, to the biggest clusters of galaxies. They all have nearly the same structure. So this is what came to be known as the universal structure of dark matter halos. However, it turns out if you go to the very smallest scales, which has only been possible recently because of waiting for the computers to get big enough to do a large enough problem, you get something different. So now we're focusing down on a really small region of space. So the distribution of matter before any structure formed was smooth on the smallest scales, smoothed out by diffusion processes in the early universe. So if you look on smaller scales, you get a smooth density structure which has peaks. And so the first things that collapse are the densest regions, which are peaks in this initial distribution of density. So here we're focusing on just one peak. And this peak is going to collapse at a redshift of 87. That means when the universe was 88 times smaller than it is today, very far in the past. And the peak, the mass in this whole region, is about Earth mass, a millionth the mass of the sun. So what you're seeing here, or we'll see when I run the movie, is 3 different projections of the mass distribution in this region, seeing it from along 3 orthogonal directions. And you can see at the time I'm starting, which is 58% of the time when it's actually going to form something, you don't really see anything. So now when we start running the movie, now maybe you can start to see some contrast here. We're up to 80% of the time until collapse, 85% of the time until collapse, 90% of the time until collapse. You can see it looks like something has formed in this direction, but that's because it's actually made a filament, and we're just looking down the length of the filament from the way I took my projections. So you can see there's no 3-dimensional collapsed object yet, even though we're 90% of the time to the collapse time. What's being shown here is the density profile I showed you before, multiplied by a power of 1.5, and you'll see why there's this r to the 1.5 in a moment. So we keep going. Oh, damn, right. Let's get to the right place. 80. Here we are, 99% of the collapse time. You can see a very strong filament, but no really condensed object in 3 dimensions. And now watch what happens. This is the density profile now. You can see there's a sharp peak here. That's this caustic corresponding to the edge of the filament. Nothing in the middle. Run it again. 96. Okay, 3% after the collapse time, there's a clear knot in the center. You see here? So an object has formed in just that last couple of percent of the entire age of the system. If you watch what happens when it keeps going, if I manage to do with this, you can see what happens. It forms something where the density as a function of radius is almost flat. So flat in this plot means it would have to be a power law. Okay, let's give up on that and look at the summary here. So what happens is as the peak collapses, almost instantaneously there's a lump at the center, which is very dense. And this lump has about the mass that was in the initial peak that we started with. And so we call this prompt cusp. This dense structure formed promptly. And it's very different from the formation that I showed you before in the Milky Way, where you saw lots and lots of things falling together and gradually building up over time. That's not what happened here. This thing fell almost instantaneously as the first object collapsed. And it formed a profile which was not a gently curving profile like I showed you for the Milky Way halo, but was a power law. So it forms in a different way, has a different structure. So if we now show the density profile, let me just show you this in the same way. This is density logarithmically against radius logarithmically. This is something which collapsed at 87. So very shortly after that, the redshift has dropped a little bit to 85. You can see the inner region here, the densest region, is following this power law of r to the minus 1.5. If you come back a little bit later and the redshift has dropped to 73, you can see it extends the power law out to this point. If you wait a little bit longer, another factor of 2 in the expansion of the universe, we get down to 37. And you can see the shape has changed. And now it looks like this dotted line, which is the universal profile. So there's a clear difference between the initial collapse of this object and the profile. So why should we care about these prompt cusps? I showed you they form almost instantaneously as each peak collapses in the initial density field. The density is a power law in radius. So you can write it as in this formula. And the power law holds between an inner radius and an outer radius. The outer radius and this constant are just dependent on the shape of the peak in the density field which collapsed. The inner radius, that's interesting. There is an inner radius. It doesn't go as a power law all the way to 0. And that depends on the nature of the dark matter, what the dark matter is actually made of, the kind of particle it has. So if we could ever see these, we would know something about the nature of the dark matter. As I showed you, the more massive objects build up like the Milky Way halo did. They build up gradually by merging and accretion, and they give rise to something which is a different shape. Now, what's interesting about these prompt cusps is because they form very early and they collapse very sharply, they're very dense. And if they're very dense, they can survive through all the later stages of evolution. They don't get tidally disrupted by the gravitational fields of the larger objects because they're so dense, they're like cannonballs. So this means that they should still be around today, even though they formed in the early universe. And if you ask how many of them there should be, well, in the standard kind of cold dark matter model which I've been showing you, where the dark matter is thought to be some kind of weakly interacting massive particle, then there would be of order Earth mass. If you ask how many of them there would be in the galaxy, our galaxy, there would be about 1 million times as many Earth mass prompt cusps as there are planets. Maybe a little less, 10 to the 5 times. They'd be mostly outside the region where the sun is, but they would be the most common Earth mass objects in our galaxy. They would have the mass of the Earth, but they'd be the size of the whole solar system. So really still quite low density. And could we ever see them? Well, that turns out to be difficult because it's low mass compared to other objects and not very extended, so it causes very little gravitational effects. But there is one way that it might be possible to see these, because they're so dense that if the dark matter particle can annihilate, which these kind of WIMPs are thought to do. That means when two dark matter particles hit each other head on, they just churn into a burst of radiation. This could be detected by telescopes as gamma rays. So one of the ways that people hope to be able to find dark matter is by looking for gamma rays from this annihilation. If dark matter really is this kind of particle, then you can ask, where would the annihilation come from? Well, particles annihilate where they can meet other particles, so it's the densest regions of space. And the densest regions of space are these prompt cusps. So when you calculate it, you find that these prompt cusps would entirely dominate the gamma ray radiation coming from astronomical sources. And so that changes the exact way in which you should look for them. So far, this hasn't been seen. But maybe it will be. And then maybe this will be the way we'll see the dark matter. And maybe these computer simulations will actually turn out to have told us where to look for the dark matter. So I'll stop there.
Thank you so much, Professor White. Thank you. It was a very inspiring sharing, and we look forward to interacting more with you in the panel discussion session later on. Thank you so much. Thank you. Once again, a big hand to Professor White. Well, up next, ladies and gentlemen, it gives us great pleasure to invite on stage Professor Peter Higeman, 2020 Shaw Laureate in Life Science and Medicine, and Hertie Professor for Biophysics and Neuroscience at Humboldt University of Berlin, to share on the topic of Opto Berlin, Shaping Biology with Light. Professor Higeman, please.
So, ladies and gentlemen, I'm very grateful to the organizers for this superb arrangement here, and I like to go into a little different direction because I don't want to talk so much about my own science. I want to talk about Berlin itself as a hotspot for optobiology and to present a few breakthroughs of completely different kind, and also show you why we are now trying to establish an international center for optobiology. So what you see here is, on the two sides, that light is a very interesting medium for biological studies because it can be shaped in many different directions, and nature has used light as energy source and also a source for information. And we use this for activation of biological system, for example, in the neurosciences, and also for monitoring events. So the person in Berlin who started this hype of photobiology was Heinrich Otto Warburg, and he was famous already because he discovered the respiratory proteins and got the Nobel Prize before he started to work on photosynthesis. And the interesting part is that he became famous for photosynthetic research, and he developed a number of methods, although he postulated at the end of his research two fundamentally wrong hypotheses. The first wrong hypothesis was production of oxygen molecules in photosynthesis require 3 photons. In reality, it needs 8 or 10. And this is based on a complete misunderstanding. The second wrong conclusion was oxygen is a reaction product of CO2. In reality, it is made from water. And you might be surprised why this person became famous. So the real reaction is water plus CO2 is converted into carbohydrates and oxygen, and this is called oxygenetic photosynthesis. They have developed already 2.6 billion years ago. So a long time for biological systems. It has been developed in cyanobacteria. It has moved up the algae world, and 500 million years is established in plants. But the question was how are the details about these complex reactions, because this is only known biochemical reaction that is able to oxidize water, which is not trivial. And that was a question that was up in biology for about 50 years before it could be solved. And the question is, why was it so difficult? So after about several hundreds of publications, the system got stuck because the molecular information about the reaction centers could not be solved. And then suddenly, in the year 2001, both structures of the photosystems have been solved in Berlin. And I focus on the second one, and that was based on the work of Atina Tsuni, one of my colleagues, who was able to produce crystals of the photosystem too. And here you see the amazing system with the manganese complex, which is the place where this so-called most important reaction on this planet, which is the conversion of water into oxygen, which is all the oxygen we are breathing in all the time, takes place. And it took another 10 years to improve that resolution that was done by a Japanese group to understand this huge molecular complex. And this is a conversion of many different proteins and hundreds of chlorophylls and carotenoids. But the center is a manganese complex, which is ultra-sensitive to X-ray radiation, so it was still a big problem to be solved. And then only this year, the details about the electron transfer have been solved, and the fixation of the reactive intermediates after abstraction of an electron. I don't know how to show anything. Maybe can do it here, and the release of the oxygen at the end. That was a major breakthrough. So a breakthrough of completely different kind was also occurring in Berlin, and that was done by George Ward, and he studied vision in the laboratory of Otto Warburg. And 20 years later, after Otto Warburg started his research on photosynthesis, George Ward recorded the absorption spectrum of the visual pigment that was done before by other groups. And what he did is, a very simple experiment, he extracted the chromophore, which was not even known that it was a chromophore, so organic molecules from the retina, and he found that they still absorb light. And this is the spectrum of the chloroform extract, and he identified this as vitamin A. So the breakthrough was, in principle, simple. Extract the retina and identify the organic molecule, which was the vitamin A, which is a big bang at that time, so that a vitamin, which was known, is the light-sensitive component of our vision. And then in Berlin, it was studied, this protein, over many decades, mainly by Klaus-Peter Hoffmann. Here's the protein, and here you see the chromophore, and here are the interacting signaling proteins that are G proteins. The next big thing in photobiology, again 20 years later, so 33 were the big breakthroughs, 53 George Ward, 73 bacteriorhodopsin. And this was interesting because this is sensory rhodopsin related to visual rhodopsin, but it transports protons, what Dieter has shown, and this was a fundamental protein for biophysical research. And at the end of many studies, most of them are done in Berlin, we understood how this molecule works. And you see here it's a shift base. Now it's illuminated. This is a proton that should be transported. And we see here an isomerization of the chromophore, so very little change in the protein. And then this proton should be released to this acceptor. One proton is released to the medium. On the other side, a proton is reconstituting the chromophore. The chromophore is back isomerizing, and then this proton is released to this water cluster. So all these little details have been studied over the years on this very principal biological object. Next problem. This is our research. It's based on observations from Minsin he did 150 years ago. So remember, 100 years it needed to understand photosynthesis. This is even longer. So in 1860, and he reported an interesting experiment. He used a suspension from the Neva River in St. Petersburg and observed these algae at high light when this was covered with a kitchen board, and they accumulated on the border between light and darkness. And in the evening, he repeated the same experiment. He observed that the cells are swimming towards the light. And the reason is I use this phototactic system to accommodate with to adjust the light intensities which are optimal for photosynthesis. So we studied the system for quite a while and realized that electrical processes are responsible for this. And we used suction pipette technique and found that depolarization is needed, and the photocounts are ultra fast. And when we used, when we recorded this at different light intensities, we found that this must be the rhodopsin. And over the years, we concluded that the rhodopsin and the ion channel must be directly linked. And then it took us another 5 to 10 years to isolate the protein, which is shown here, and we reconciled it together with my co-laureate Georg Nagel in xenopus oocyte, and we found photocounts that are very similar to the photocount in chlamydomonas. So this was the first breakthrough in our field. No, the second, yeah. So the first was recording of photocounts in chlamydomonas, and here the discovery of the molecular protein. And another key experiment on this line was the expression in human embryo kidney cells. So the fact that this protein also works in human cells. And this triggered certainly the expression of this protein in many other cells. And this was the birth of the field called optogenetics. And here's an experiment of Carl Dijsselrood, and he expressed it in neurons, and if you apply a train of light stimuli, you get responses of action potentials. So the principle of optogenetics is very simple. You take the DNA, you take the DNA from an algae and pack it into a virus together with control elements of the target cell, and then you transfect eyes, heart cells, different brain cells, or plants, and then you can express it in individual cells that understand the regulational element, and you can fire action potentials or study the function of these systems. So then you can study certainly different modalities of the human behavior, like learning, feeding, motion, vision, hearing, sexuality, aggression, autism, addiction, anxiety, and many more, Parkinson, and so on. So then we modulated the cell in many different, the protein in many different directions, made it slower or faster or redshifted, or make it bimodally activatable and inactivatable. And then we could study the brain in many directions. One example is here. Dopaminergic cells have been transfected, and here you see the response of a normal brain, and after inhibition with an inhibitory channel rhodopsin, you can strongly suppress the response. Another experiment is to identify individual cells for different modality, and one experiment was done with this super slow channel rhodopsin, and what we see here, we can activate we can activate the cell with one flash, then the cells remain depolarized for a long time, and then you can deactivate it with a different light quality. And we expressed it in sub-regions of the prefrontal cortex, where the neuroscientists expected to find the cells for anxiety. And if these cells, if the mouse who is expressing this super slow channel rhodopsin is exposed to a maze, then the mouse is only using this covered arms. But after depolarization of the anxiety cells, it's exposed everywhere. So light is blocking anxiety if you stimulate this very specific cells of the brain. So, and the last application is shown here, or the last spectacular application is here, where Sahel in Paris and Boto and Roska developed a system to recover vision in humans. So when the cones, rods and cones are degenerated as a response of retinitis pigmentosa, and they expressed a channel rhodopsin from green algae in these secondary cells. Vision could be recovered, and here's an experiment, or here's a person who has been treated with channel rhodopsin, could at least identify objects, can move around, can see stripes on the street, can identify people, and can at least identify objects that are necessary for their daily life. So medical application is in sight, and we are very certainly happy that this is possible with an algal protein from the ocean or somewhere else. So the question is why we need a new optogenetic center, and the reason is that we are not done yet. So our interest was to establish an international center for optobiology to integrate optobiologists from Berlin and other countries. And the question is what remains to be solved, and here are a few examples. So one example is magnetoreception. This is a sense of birds which has not been understood despite many nature publications in the past 10 years. It's absolutely unclear how the birds are doing is to orient at the magnetic field of microtesla. And another one is flowering. We don't know how flowering is controlled. We know flowering is only possible during a certain day length and a certain temperature, but how these two modalities are combined is unclear. And we don't understand the high order or symmetry of the flowers. The next topic we would like to address is UV and infrared vision, because it was thought for a long time that rhodopsin only work in a certain spectral range that we cover with our eyes, but this is not the case, and the UV receptors, for example, in this shrimp, mantid shrimp animal, the UV sensitivity in this range and the infrared, near-infrared sensitivity in this range are completely ununderstood, and we know that the principle of the excited state reactions is completely unclear. Another application is the cardio-optogenetics. So there are strong activations, and cardio-optogenetics can be understood in certain ways, either to control optogenetically the communication between the brain and the heart, and the second is the direct activation of the heart, so to generate optogenetic pacemaker. And the third application is to cultivate different heart cells and then optogenetically activate them and test cardiotoxicity or effects of drugs for personalized therapy. So many applications are possible, and another aspect is we need certainly artificial intelligence-based functional genomics, and the reason is that hundreds or thousands of genes come to the market, and in the past 5 years, what you see here is about 1,000 identified channel rhodopsin genes have been found, and the question is, how can we study them? We cannot study them individually, so we need computer systems to predict the function more precisely. And also completely new rhodopsins or light receptors have been discovered, for example, enzyme rhodopsins that we studied in the past years with an enzymatic N-tail, which is or enzymatic unit, which is light activated by transfer of information from the rhodopsin part to the enzymatic part. And on the other hand, also very bizarre or unusual photoreceptors like this best rhodopsins that has been identified somewhere in Hawaii by this Tara Ocean boat, which takes samples all over the ocean. And this is the first tandem rhodopsin, with two rhodopsins in sequence connected to an ion channel. And this is a spectacular photoreceptor, and the structure has been recently solved by Moran Shalev at the Weizmann Institute. Here you see the spectacular structure. And it's also amazing that the absorption is far redshifted for whatever reasons. And we studied the electrical properties and found that this is a light-gated chloride channel. And we are convinced that the real function is quite different. And we think that these large channels are located in the algal chloroplast, and they are responsible for photosynthesis and to conduct CO2 in several organisms. So, last and not least, another tendency or another direction of opgenetics is a subcellular opgenetics, because we have many modalities within the cell, which is transport of molecules along microtubules. This is gene transcription and translation. This is protein degradation and all kinds of things that can be triggered optically, and this should be exploited in the near future. And here you see a cellular image where different substructures have been labeled. And for example, Johannes Vierok, who is a young research group leader at the Charité, my former student, he established opgenetics and lysosomes with his student. And what you can see here, light-induced acidification of these little lysosomes off and on, so we can study the function of these subcellular systems. So I hope I could convince you that the opgenetics or optobiology system is not at its end, and you can make completely different breakthroughs. You can solve for structure, you can start a new optical system, and I think there is plenty of room for the young scientists to go into these fields, and there are amazing discoveries to be made. So this is my group on a visit to Prague, close by, and we always had fun, although we also had to pass lag periods over decades to solve a system, but at the end it was a great pleasure to work with this young students. And what we observed over the time, the students remain the same. They are active, enthusiastic, and the only people to get older are ourselves. So thank you very much.
Thank you so much, Professor Hickerman. Thank you. Please be seated off stage. We look forward to more exchanges with you at the panel discussion session. I personally love the slide about the unsolved photobiological questions. Borrowing the words of one of the speakers this morning, any problem is an interesting problem as long as we don't know the answer yet. Right, so there are unsolved problems and interesting research work to do. Up next, I would have the honor of inviting on stage Professor Victoria Caspi, 2021 Shaw Laureate in Astronomy and Lauren Trottier Chair in Astrophysics and Cosmology, distinguished James McGill Professor in Physics of McGill University, giving a presentation on trends on radio astronomy and the CHIME telescope. Let's welcome Professor Caspi.
Thank you very much. It's a great honor and a pleasure to be here. My first time in Hong Kong and having a really, really wonderful time. And I'm going to talk to you, it's an astronomy talk about astronomy in the computing age, and give you an example of the new trend in astronomy with the CHIME telescope. So traditional astronomy, when you say astronomy, many people picture a dark night and a telescope where you point and look at your favorite object, and you choose which object you're going to look. You turn your telescope in that direction, and that's the one you look at. And even with professional grade forefront astronomy with very large telescopes like the Gemini telescope in Hawaii, it's very competitive to get time. You have to tell people where you want to point, what object you want to see, and convince them that's a really interesting position on the sky to look at. And this is true in optical astronomy, but really generally true throughout astronomy. For example, in radio astronomy as well. You see a picture there of the Murayang telescope, the Parkes Observatory in Australia. That's a telescope where you point and you choose what radio source you want to look at. Or the tremendous, most sensitive telescope that we have, radio telescope today, the FAST telescope. It's also a pointed instrument. In this case, the surface deforms and helps you point in the direction of your favorite object. Now, that makes perfect sense. I mean, this is how we build telescopes, or how we've built telescopes in the past, because we know what's out there, and we know where to look, and we know where the interesting objects are, and we can predict exactly. For example, here's some very famous stars in a constellation, Orion, that you might be familiar with, and you know that you can go out the next night, and Orion, these stars will look the same, and if not just the next night, you could go back 100 years from now and look at Orion, it's going to be the same. You can predict exactly what the sky will look like. 1,000 years, 100,000 years, it's going to look the same. That's what we've been used to. But of course, that's not really true. That's not actually true. Even 400 years ago, Galileo, when he looked up at the sky with his tiny little telescope, he could see things were changing. He could look at Jupiter and see that, you see a hand sketch that he made of the moons of Jupiter orbiting. He could see changes from night to night in the position of the moons of Jupiter. Even our own sun today, we know, is volatile and can produce solar flares and all sorts of coronal mass ejections seen here. And this is the sun as it looks from a UV telescope. It changes, it's rotating. Lots of things, in fact, change in the night sky, or the day sky. And here's a very famous object, the first object in the Messier catalog, the Crab Nebula, which was seen to be formed in July of 1054 AD. In fact, by Chinese astronomers, it was called a guest star. It's a supernova explosion of a massive star at the end of its life. This was a huge explosion. First, there was not much to be seen there, and then in 1054, it became as bright as the full moon for weeks. And today we see this spectacular nebula, which we can observe to be expanding, remnant of this huge stellar explosion. And nowadays we also know about many other types of objects that explode, different reasons for exploding. The crab is from the collapse of the core of a massive star. Here's an example of merging stars, merging neutron stars, and we've observed these kind of mergers, both optically through kilonova, as we call them, through gamma ray bursts when they explode, or even through gravitational waves now with gravitational wave detectors. These are tremendous explosions, very spectacular, but in this case, hard to predict. You don't know where the next one's going to happen. It's a fundamental problem. You need detectors that can look in many directions if you want to study many of these objects. In fact, many cosmic sources we now know are transient. You might think of the night sky as unchanging, but in fact, it changes radically. There's objects that appear and disappear, and on a wide variety of timescales. There's objects that will appear and be there for weeks to months, and then there's others that will appear and vanish in the matter of seconds, or as I'll describe, even milliseconds. So there's a revolution right now in astronomical observations. Computers, they're revolutionizing our view of the universe. These are ordinary computers that you might be using to play games, but we can use them in astronomy to study the transient sky. The universe is a highly variable place, and we call this time domain astronomy, where we look at variations in the night sky on a wide variety of timescales. And I'm going to tell you about one example of time domain astronomy and highly variable objects, fast radio bursts. This is what I study nowadays. These are mystery objects. I sometimes, people ask me, what do I work on? And I say, I don't know, because we don't know what these things are. In fact, they've captured the imagination of the popular press because it's a true astrophysical mystery, these brief flashes of radio waves all over the sky. This field started fairly recently on astronomical timescales, certainly in 2007 with what we call the Lorimer burst. And the Lorimer burst actually, the team that discovered the first fast radio burst was awarded the 2023 Shaw Prize in astronomy to good friends of mine. You can see them there, Matthew Bales, Duncan Lorimer, and Maura McLaughlin, using the Parkes telescope discovered by accident, something not predicted by any theory. They discovered this burst, which you can see here. This is the signal from the radio telescope, from an antenna that's sensitive in the frequency range of 1.2 to 1.5 gigahertz. That's on the y-axis of the plot. And on the x-axis is time. That's time going by. That's about half a second of data, just half a second of data. And what you can see here, this horizontal line is a boring television station. You can tell there's lots of problems we have in radio astronomy with terrestrial interference, something that's always on at a single frequency. It's probably a TV station or cell phone or something. The astronomical signal, though, the Lorimer burst, is this sweeping object that arrives at the high frequency first and then comes down and sweeps through the radio band. That's called dispersion. That's due to the interaction of the radio waves with interstellar or intergalactic plasma, and in fact tells us that this object is coming from very, very far away indeed. There's more dispersion, more plasma implied by the sweep of this signal than can possibly be explained by our Milky Way galaxy alone. That dispersion sweep you see there is telling us this object is coming, in fact, likely from cosmological distances. But if you remove that sweep in software by digitizing the signal both in radio frequency and in time and correct for the sweep, you see the inset, which is a burst that comes out of nowhere. There's nothing, then there's a bright burst, and then it goes away and doesn't come back. That's the fast radio burst. So what are the key properties of these objects? Like I said, they're bursts that last just a few milliseconds, and they're happening all over the sky. By a decade after the Lorimer burst was detected, astronomers had discovered roughly 24 more such objects, so detecting them at a rate of about 2 per year or so. But even with that low detection rate, with so few of these objects detected, we could tell that the inferred all-sky rate, so that if you could observe the entire sky all the time, you would detect about 1,000 of these per day. So it may be very short, but it's something in the universe that's very common. It's ubiquitous. And as I said, from the dispersion sweep that you can see in this other event that was also detected at the Parkes telescope, we can infer these events come from far outside the Milky Way galaxy, from cosmological distances, which means they have to be incredibly powerful explosions of some type, something incredibly luminous to be detectable from such large distances. So extremely powerful explosions, but we have no idea, or we had no idea what's causing them. Their nature is unknown. And, how do you study a phenomenon like this? How do you study something where you don't know where or when the next event is going to appear? That's a fundamental problem. That was a fundamental problem in the field. But in Canada, we built a new kind of radio telescope, the Canadian Hydrogen Intensity Mapping Experiment, CHIME, which was, from the title, from the name of the telescope, you can tell it was designed for something totally different. It was designed to study redshifted hydrogen gas, to study what called baryon acoustic oscillations and the accelerated expansion of the universe. Now, this doesn't look, to many people, it doesn't look like a telescope at all. This is made of 4 cylinders, each 100 meters by 20 meters. This is a very large structure. 4 cylinders, and they're oriented north-south, and there's no moving parts to this telescope. You cannot point it where you want. It only looks directly overhead, but because it's oriented north-south, the sky drifts overhead as the Earth rotates, so that every day we can see the entire northern sky. It's sensitive in the frequency range of 400 to 800 megahertz thanks to 256 antennas that are hanging on the axis of each of the cylinders, for a total of 1,024 antennas that are all looking up at the sky, each with dual polarization available, so a total of 2,048 input signals. The total data rate that we collect at the CHIME telescope is approximately 13 terabits per second. The data rate that's comparable to the entire world's cellular network. It's all being analyzed in real time on the CHIME site, as I'll describe. Just to set the scale here, it's a very large structure. That's our team, lots of energetic, wonderful graduate students from my research group and from other groups that we worked with from University of Toronto, University of British Columbia, and also, of course, McGill University, where I'm from. The total collecting area of the telescope in commonly used Canadian units is approximately 5 hockey arenas. Very proud of that. Why would you build cylinders? Why cylinders? Well, conventional radio telescopes like the Parkes telescope see a tiny piece of the sky at one time, a very small field of view. And so if you're interested in transient sources where you don't know when the next one's going to come, you're going to miss most of the sky. You could have a huge transient event going off over there, but you're pointed in the wrong direction. Now, when you have a cylinder, it focuses only in one dimension. The radio waves in the opposite direction, or the cylinder is like a mirror. It doesn't focus it at all. So what you see is a large swath across the sky with a total field of view that's 2 orders of magnitude, at least, greater than that of the conventional radio telescope. And so if you compare the total field of view of a conventional radio telescope with that of the cylindrical design, you can see that simply put, you're going to just by sheer field of view detect many, many more of these transient objects. And that's what makes the CHIME telescope, designed for totally different purposes, so great at studying fast radio bursts. Now, if you look at the bottom there, if you put a single antenna hanging on the cylinder, you'll see indeed a large region of the sky, but if you detect something, you're not going to know where it's coming from. You see everything equally in that area of the sky. But if you put many antennas and then you feed all of those signals into a powerful correlator that adds in delays, so you treat it as an interferometer, it adds in delays so that you effectively digitally see many independent positions on the sky at once, then you can tell where in which sky beam the signal originated. And then if you populate all 4 cylinders, you can see where in that main primary beam, both in the north-south direction and in the east-west direction, you're effectively sensitive to many, many different positions on the sky all at once. And so this is a cartoon, but this is actually what we do, digitally form 1,000 different beams, all pointing in different positions on the sky all at once. So observing with the CHIME telescope is like having 1,000 Parkes telescopes all operating at the same time, We have effectively 1,000 telescopes able to search the sky for fast radio bursts all at once. Now, I said that's for fast radio bursts. I mentioned that the telescope was designed to study neutral hydrogen, highly redshifted neutral hydrogen, to study the expansion of the universe. And it can do both experiments at the same time and much more, all at once. So this is a signal path. Here are the cylinders. It goes into an FX-style correlator, where the first part of our correlator is based on field programmable gate arrays built by my colleague at McGill University, Matt Dobbs. And then that's called the F engine. The X engine is based on GPUs. We have 256 GPUs that sit in huts. I'll show you shortly underneath the telescope, built by my colleague at University of Toronto, Keith Vanderlyn. And these signals are fed into several different backends. Here is the CHIME FRB backend that does the FRB searching. By taking that signal, the 1,024 stationary beams on the sky that are being read out every millisecond to search for fast radio bursts, at the same time as the cosmology backend is studying spectra all over the sky. And even we can actually study, we form 10 digital tracking beams on the sky. We can observe 10 pulsars in our choice anywhere in the primary beam at the same time, 10 radio pulsars. So you can do, and actually you can do many things. We're planning pulsar searches, studies of absorbing systems. There's many different types of science you can do with this digital radio telescope all at the same time. So I said all of this incredible data rate is processed on the site. The F engine is sitting in huts that are directly located under the telescope, and the X engine is in here. And you could see here our signal path. This is a trigger experiment. We can't possibly record all of this data. For the FRB experiment, we are searching in real time all of this data, and we buffer the data in two different buffers, one that's intensity data and one that's raw voltage data. We only dump those buffers when we detect a signal, and that means if we miss a signal, we've lost it forever. So we have to be very, very careful. Now here was how many fast radio bursts were detected prior to CHIME. You see many of them were at the Parkes telescope and at the ASCAP telescope in Australia. And when CHIME turned on, you can see there's a bit of a difference there. We started detecting lots of sources, not at a rate of 2 per year like prior to 2017. Now we detect them at a rate of 3 per day. And that's continued on for several years now. You can see here the CHIME telescope, when we started detecting FRBs, it was recognized for this kind of work, and you can see lots and lots of fast radio bursts that we've been detecting. And in this field, it sometimes feels a little overwhelming. You can see what it's like in CHIME domain astrophysics. The FRBs don't stop coming, and we feel sometimes quite overwhelmed. And I think that's actually a theme in time domain astrophysics and the future of astronomy. Tremendously data-driven, more data than we have to figure out how to handle all that kind of data. And so what are some of the discoveries we've made very quickly? We have discovered that there's some fast radio bursts that repeat. This was actually discovered, the first one was found by the Arecibo telescope, and we found 50 more of these repeating fast radio bursts. Small fraction, only about 3% of FRBs are seen to repeat. And here you can see that they have different emission properties. This is showing width on the x-axis, and this is a property of the spectral properties of fast radio bursts. You can see the green ones are the non-repeaters, the orange ones are repeaters, and they have different temporal and spectral properties, which suggest perhaps fast radio bursts, there's multiple classes, maybe. We don't know yet. We've even discovered one repeater that likes to burst only every 16 days. Very strange. It caught us very by surprise. You can see this is a period. You see the spike at 16 days. This is the exposure of the telescope. We have lots of exposure, but the bursts only come in 4-day windows. every 16 days, and we don't know why. That's a very curious source. It's when you discover lots of sources, you can make many interesting discoveries. This is one of now we have thousands of FRBs. One of them, you can see just a couple milliseconds of data. This is our baseband data, very high time resolution. You see spikes, some of the spikes in here, this amazing source last only a few microseconds. And the amazing thing about this sort of technology, using computers in this way, is that it's very scalable. So as computers get faster, we can replace the correlator, we can replace the search engine that finds fast radio bursts with ever faster computers. And soon, we can just start looking at the microsecond sky, because we see some of the FRBs have microsecond features. Perhaps there's way more objects in the universe that last only a few microseconds, and we don't yet have sensitivity to them, but we could very easily, just by scaling up the computing technology we use now. So in summary, the time domain astronomy is revolutionizing our view of the universe. CHIME's enabling huge progress on millisecond timescales, but I think soon we could even hope to start seeing the microsecond universe with CHIME and with other telescopes. Thank you very much.
Thank you very much, Professor Caspi. If we could invite you to please remain on stage and be seated for the Q&A panel session. Well, I think you would agree with me that the three laureates' presentation has unearthed a lot of the latest advancements that have a link to new technology. And up next is time for you to participate as well, because we're about to have a Q&A panel discussion. You could either raise your hands for questions for our panelists or utilize the app, HKLF app, in the Q&A function to pose questions. Before we begin the session, I would have the honor of introducing you to our panel chair, Professor Wang Yang. Professor Wang is the Vice President for Institutional Advancement and Chair Professor of Department of Industrial Engineering and Decision Analytics and Department of Physics, Department of Mathematics, Director of HKUST, the Big Data for Biointelligence Laboratory of the Hong Kong University of Science and Technology, a huge affiliation because I graduated from HKUST as well. So ladies and gentlemen, let's put our hands together for our moderator for the session, Professor Wang, please, to join Professor Caspi on stage. And once again, can we have on stage Professor Higeman and Professor White once again? Thank you, gentlemen. All right, now that the stage is all set, Professor Wang, the floor is yours.
Okay, so it's a tremendous honor to be the moderator of this session with three extremely distinguished scholars. And we don't actually have much time, so I'm just going to ask maybe a couple questions from each one of you, then we open up the floor for everybody. And I'm going to begin with Professor White. So, you know, I listened to your talk with a tremendous amount of interest. And your research focus, you talk about the finding structures in this vast universe, right? And you have done a lot of simulation, and these figures look very fascinating. So it also reminds me, you know, a different area, the molecular dynamics and protein folding. So also, at one time, we've been using a lot of large computing power to see how this folds. But recently, we have also seen, deep minds using AI technology to create AlphaFold, which also vastly increases the efficiency, but uses a lot of data, of course. So how do you see this? Is there like a similarity? Do you see your research in the area also going in that direction?
I think there's a problem that's general to many areas. So I like to compare with climate or with macroeconomics or with perhaps protein folding would be another example, that we can now be looking at very complex systems with very numbers of degrees of freedom, which we can observe partial pictures of through different techniques. And we now have very powerful computers which can simulate also very complex systems which produce extremely complex pictures. And the question is, how do you learn something which deepens our understanding of systems by comparing these two? Because our observations, whether of proteins or of the universe, are always partial. We're sensitive to the aspects that are observable, which may not be all of the aspects. And the simulations, although they're, I won't say equally complex, but also extremely complex, they're also only partial realizations because we're unsure that we're modeling all of the physical mechanisms or the biological mechanisms which are relevant. And so when you get an agreement, this could be by chance because you changed your model, changed the premise to get agreement. And so how do we know what we've really learned from comparing a complex but incomplete simulation with a partial observation of a complex system? And I think all these areas, this is a challenge. And some people say we should compare things by machine learning. My point of view is then we understand even less about what the machine has picked out. Of course, the machine learning is very good at pattern recognition, but we don't know what the pattern is that is found in common between our simulation and the real world. So I think this is a whole area of basically the epistemology of research. How do we get knowledge out of comparing these kinds of systems?
I see. So actually, just a follow-up. Right now, I mean, you mentioned that you can now go down to scales that are much smaller than used to be because of computational power. You mentioned the increase of computing power about 10 to the 9th or 10 to the 10th. In your research, what kind of, how much computing power do you actually use?
Well, in my field, since I would say about 2000, we've always done computations lasting months on the largest available computers. And so we actually are the people the supercomputer centers love, because as soon as they get a new machine, we can use a large fraction of it and then tell them it's not big enough, which is the justification they need to buy an even bigger machine. So in practice, we can use a lot, although this trend is changing because what we need is rapid calculation. We also need very large memory, which probably would be for Professor Caspi also relevant because you want to store a large amount of data. And in recent years, the trend in computation has been to concentrate on the speed, using GPUs, for example, as we heard. They have a characteristic of typically having less addressable memory per processor. So actually, for many purposes, it's difficult for us because we need both the rapid speed and the large memory per processor. So more modern computers are in some ways are less suitable.
Yeah. Okay, thank you. Peter, I also want to ask you a question. So you are obviously a renowned neuroscientist, and so you talk about, I mean, you pioneered this, the photogenetics, and you're obviously have delved deeply into this field. What do you, is there, research-wise, but now I want to ask you about treatment, okay? Do you see the potential of photogenetics in the treatment of neurological disorders?
I think what I showed today is that treatment is possible in cases where the organ of defect is light accessible, and therefore the eye is certainly the organ of choice. So to treat eye defects is certainly much easier than to cure other defects. So another application which is quite well developed is the treatment of deafness. And then you can express light-sensitive proteins in the secondary cells of the inner ear, because the inner ear is nicely accessible by light as well. So you convert sound into light and then illuminate these cells with incorporated light-sensitive proteins. Other possibilities are to treat Parkinson, because the responsible cells are very localized in the hypothalamus, and this is accessible by short light guides. Other problems, defects, where cells are involved that are widely distributed over the prefrontal cortex are not easily accessible because the brain is dark, and to illuminate a wide range is not possible. So optogenetics is first of all an analytical instrument, and treatment of patients will remain an exception. But a few cases are in the pipeline of clinical trials.
I see, yeah. Actually, just a follow-up. So, for neuroscience, biological research, we spend a lot of time doing experiments in labs, and sometimes, data, of course, it's difficult to obtain a tremendous amount of data. But do you see also, since today our topic is trend in, new technology trending in scientific development, do you see, you've been using a lot of computation anyway, so do you see, AI, for example, playing a major role in the future research of this area?
Yeah, I briefly touched this point. The problem is we get information of new genes over and over. So we get, probably these days, a couple of hundred channel adoptions, but no one is willing or able to test them all.
Yeah.
So we need an artificial intelligence systems as an intermediate device. So it has been we have to feed them with information that is there already, and then to analyze new sequences, structurally wise and functionally wise, with artificial intelligence could distill the proteins that are of real interest. And this is a potential. To predict novel proteins has not been possible with artificial intelligence, but to analyze better genes of the type we already have is certainly unavoidable, I would say.
Yeah, okay, thank you. Professor Caspi, you have obviously been, I was very fascinated about the talk you have, the fast radio pulses. And so you also understand you have been an expert in neutron stars, for example, okay? So how do these, for example, what's the importance of these in terms of shaping our understanding of the universe?
The importance of neutron stars or of fast radio
both
bursts?
of them, yeah.
Well, I'll start with fast radio bursts. So,
Yeah.
I presented it as a puzzle that, unpredicted and a phenomenon we don't really understand. And, what we're trying to do is understand the mystery of what these things are. But that's actually really, I didn't have enough time, but that's only half the story. Because when you have these bursts of radio waves that you could see across the universe, they are actually, even if we never figure out what they are, even if it'll always be a mystery, which I hope is not true, but even if you never figure out what they are, they're tremendous probes of the matter through which the radio waves propagate. So radio waves get imprinted with information, detailed information about the matter that they went through. So for example, some of the FRBs we detect have scattering tails because they've been scattered by density inhomogeneities that result in slight delays of the radio waves. So we see these scattering tails, and we can tell something about the distribution of matter through which they've propagated. We get the dispersion, the amount of dispersion that we see that tells us also about how much plasma they've gone through. They scintillate. We have they twinkle in radio waves. That teaches us about the motions of the plasma in homogeneities through which they propagate, and they're even polarized. Some of them are highly linearly polarized, and we can, through Faraday rotation of the radio waves, we can determine information about the magneto-ionic properties, something about the magnetic fields through which they propagate. So by studying fast radio bursts, we have a truly novel probe of the matter distribution. In fact, we can test some of the models that Simon presented about halos of galaxies by looking at fast radio bursts. that propagate, whose line of sight is very close to galaxy halos.
I see.
Yeah. There's also neutron stars, but that's a whole other...
Yeah, that's a whole new...
That's a whole other thing, yeah.
I'm always fascinated about this kind of, research because my understanding, in fact, I look at your pictures, you have a lot of noise, right, when you receive these signals.
We have a lot, sorry.
Noise.
Yes, yes.
And you even, mentioned you don't know where, sometimes you don't even know where they come from.
Yeah.
So this must be a tremendous challenge. What do you see in terms of, the scientific development? You also mentioned the evolution of instruments, right, time domain, evolution. And mentioned that in 1500s even, people observed this, this big explosion in the cosmos.
Yeah.
But now technology has greatly expanded, advanced, right? What do you see the future of, what kind of technology are going to emerge in the study of astronomy in the next phase or next few years?
Well, I work in radio astronomy, but the time domain and large survey-type telescopes are going to be coming online. For example, the Vera Rubin Observatory, the LSST, will revolutionize, in the optical domain. And I think, as we have the more technological ability to see huge areas of the sky repeatedly over and over again, we're going to see many transient sources, and then we'll want to correlate all these different data sets to see, for example, a question you might ask about fast radio bursts is there an optical burst there? Is there an optical transient there? Or perhaps there was an optical transient a year before the radio burst, or when? So we're going to have more and more data about the sky and its variability, and I think that will be the future. And you were talking about AI. I think AI it's already very important in astronomy, and certainly we use it to filter out noise in the chime pipeline. But that will be a way to bring these different types of data sets together and perhaps identify multi-wavelength type of new transient sources.
Okay, thank you. Simon, actually, the one thing I want to ask you is perhaps in the, today when we talk about new technology, we also talk about the technology impacting of research and education, right? Research and education, in terms of new technology, especially AI technology, for example, and other kind of technology. So how do you see the new technology impact our research, for example? In terms of research discovery, are we going to rely more and more on computers in the future? Can they, for example, help us even discover new things autonomously?
Well, I think we will undoubtedly rely more and more on computers for everything. This is inevitable. Whether they help us understand more is perhaps debatable. And that I don't know. AI is very good for pattern recognition.
Yeah.
It's very good for interpolation on very high dimensional space. Extracting the physical basis for phenomena, it's yet to be demonstrated, I think, whether this will work. I think it's an open problem in AI research itself, is how to extend the interface to what we normally understand by scientific, the growth of scientific understanding of phenomena. I think it will help us because it should help us to see the patterns. We still need to interpret the patterns in terms of some theoretical understanding of what is generated and why they have the shape they do. And that aspect, why things look the way they do, I'm not sure that the computers can replace the people yet.
Yes. Peter, you actually want to say something about this?
Yeah, I want to say something. We also use more and more computers, but what I have to tell the students that big data is not big science.
Yeah.
So it can help certainly to analyze data, but in many cases, the data are the most interesting ones that cannot be analyzed by AI. So that not fit into the current system, that not can be explained by the current knowledge we have about the system, and the students need to focus on those to make new discoveries. This is my message. So don't focus too much on AI, but focus on those data that do not fit.
Yeah, okay.
I agree wholeheartedly with that. I think you have to focus on scientific understanding, and and it's the same as you shouldn't worry about whether a problem is important, but whether it's interesting. We keep hearing this, and I think this is the most critical message.
Yeah, I see Victoria eager to jump in. Okay.
Well, I mean, I guess I'm a little, more optimistic. So I think I agree with everything that's been said, but I think there's tremendous potential for AI. So I think about my field, I showed these crazy bursts, but there's fundamental expectations based on physics. So you might expect, for example, that there should be a width-distance correlation for fast radio bursts. If they're formed in a distant universe at a time when the scale factor was much smaller, different, then you should see cosmological changes depending on how far away they are. Now, to fit the widths out of these crazy bursts, so how are you going to do that? We sit there and we try and do it by hand. When you have thousands of bursts, and that's the size of the population you need, you don't want. I think it's boring for a student to have to sit there do it, but what's interesting for the student is to find the algorithms, and I think that will absolutely incorporate AI, that will do it for them, and then be able to reveal the interesting correlations that are in these large data sets. That will be very, very illuminating of the physics and cosmology. So
I see.
I think there's potential.
Thank you. Actually, I want to open up the floor, the panel to the floor for questions. But I also want to comment on this. I think right now we have seen that the OpenAI model, GPT-4, for example, can solve some simple math problems. Sometimes actually not so simple math, but still they're very far away from discovering important theorems in mathematics. So for that matter, I think my job is still pretty safe, okay?
But the gradient, how fast it's changing.
Yeah, it is approaching very fast, yeah. Okay, let's take some questions from the audience. Yes, I see one. Can we pass some microphone? Do we have microphone? Yeah, we have so many people asking questions.
This is Jiwon from Korea. I would like to thank you all the professors for the interesting talks and the panel discussion. I would like to ask a question for Professor Victoria. In the last slide, you mentioned about scaling up, and I would like to ask you about the future. So if there would be a chance of expansion of your facilities, what would be your priority for your next step? Maybe splitting time more, or doubling the receivers of the telescopes, or having another set of telescope in the southern sky, or maybe others. So, and how would it benefit your current research?
Yeah, thank you very much for that question. And everything you suggested is possible. So in fact, what we have prioritized is many different options. And part of the challenge is choosing which one to do first. So what we have chosen to do, CHIME has been tremendous at detecting large numbers of these objects, but what it doesn't do very well is localize them on the sky. So if you want to know what galaxy it came from, with CHIME, it's very hard to do that. And that's simply because the radio telescope's angular resolution is not very good. So what we have decided to do is build several more CHIME telescopes around North America, all to look in the same place as CHIME does, and to buffer data, and to use it as a very long baseline interferometer. that these additional CHIME telescopes, that we call them outrigger telescopes, will dump their data upon a trigger from CHIME, and then we will correlate the data offline and determine, we plan with several thousand kilometer baseline separations between these telescopes. Interferometrically, we can determine to tens of milli-arc seconds, and that will tell us not only what galaxy each one of the CHIME FRVs has come from, but where in the galaxy. Does it come from near the center of the galaxy? Does it come from the edge of a galaxy? Does it come from a star-forming region? Does it come from a globular cluster? That's what we've planned to do first, but we could do many other things. There's only 24 hours in the day. Yeah.
Okay, another question? Yes, this lady here.
Thank you for this interesting session. I'm Chitra Joshi from University of Oxford. I have a biochemical question to Professor Peter Hegman. So you mentioned that the optogenetics has a limitation regarding accessibility by the light. I wonder if you have tried integrating the luciferase system with opsins, where you can administer luciferin and activate the optogenetics system. And if you think this can be done, do you think there is an opportunity to develop this maybe system as a biosensors for neurodegenerative diseases, for example, like Alzheimer's, where it is about the accumulation of misfolded proteins? So can we target those developing a biosensor and understand?
Yeah, this is an interesting question, because I never believed that this system will work before I've seen that my colleague, I forgot the name unfortunately, linked a luciferase to the channel rhodopsin and could use the light of the luciferase, for example, upon calcium influx into the cell, to activate the channel rhodopsin. But this is for a different purpose. This is for monitoring different events in different cells. So to directly activate channel rhodopsins via light certainly provides a much better time resolution, and for different purposes it can not so easily done in widely distributed cells simultaneously. But certainly the technology you're mentioning is very interesting for monitoring. But on the other hand, long-term activation distributed over the brain can be, in most cases, better done by chemical drugs, because you can apply them through the bloodstream, and the advantage of optogenetics on this long time scales is not as great anymore.
Okay, another question. By the way, you can also submit questions online, and I'll see instantly your question on my iPad. Another question? Yeah, we see someone raising. Yes.
Thank you. I have a comment. I don't have a question. Is this okay? Because the discussion about AI is super interesting, and I have a comment on this. And it sounds like the conclusion is AI is very useful. I agree with this, but I'm not optimistic on that AI can advance the frontier of science. And because the reasoning is as follows, because there's a book by an Israeli writer called The History of Sapiens. So, and he has a very interesting argument that there are more than one species of human beings, but somehow the sapiens is able to conquer other species and survive until today. And the reason is because sapiens is incredibly good at imagining something which not exist in this world, and we are incredibly good at conceptualize things. And for example, the dark matter, and this, this could be an illusion, it could be a fallacy, which doesn't exist in the world, but somehow we make up a concept called dark matter, and we are able to study it. And I agree that AI is useful, but only in the sense, so like you said, that you are already use it to filter out noise, but that's because it's a large amount of work, because we cannot manage to do that large amount of work. So, and the AI's computer can do that, right? So if you have a super big computer. But I think that is contrary to the essence of intelligence. We are good at simplify things, not human beings are not good at do complicated things, but I think, so I think in the future, I think if AI is able to advance the frontier of science, that's not because the AI is good at doing a super big work, but that's because we finally understand the essence of intelligence. So that's my comment for this panel, the discussion of this panel.
Yeah. Thank you. Thank you. Thank you for your comment. I think what you mentioned is very interesting, and it will probably take us another day to, to go through everything. But I do receive a question, for an online question for Victoria. Oh, you want? Yeah.
Well, I was just going to say that I read Harari's book too, and I think it was very thought-provoking. I don't think he proved all of his points, but one of the things that people say distinguishes people from other is our ability to develop and use tools and to make new tools. So my point of view is that AI and indeed the computers should be used as new tools, and we're still learning how to use them. But I don't think they're going to, some people are worried about general artificial intelligence taking over from us. Perhaps this is a threat, but I don't see this at the moment. For me, these are still tools which we get to use.
Anyone of you who want to chime in on this comment, or? Okay. So actually, I do receive a question online for you, Victoria. Actually, it's also related to my question to you. If FRB were observed haphazardly, how does one identify that such structured noise is actually a new signal? Is it possible that other novel effects could be found by revising old data? Might this sort of revisionist survey lend itself well to an ML AI assisted search? What are your and perhaps other people's best guesses as to the origin of FRBs?
Yeah, that's a lot of questions. But so the first, absolutely. So it was not. It was discovered in sort of by accident, but the idea was some galactic neutron stars show individual pulses that are quite bright. And so the team that found them was looking for sources inside the galaxy and looking for individual pulses. And now they didn't expect this size of dispersion in the direction they were looking. So in that sense, it was not totally haphazard, because they were actually looking for bursts of radio waves. They just weren't looking for ones that could possibly come from outside the galaxy. We didn't think that those can exist. And so absolutely, you could go back through old data and then look for more of these. In fact, they found more that way, but that's limited because before, computers weren't as fast, and so the cadence of the data, you need millisecond or faster time resolution to find that. So if you don't have that in the recorded data, you can't. Yeah. And the best guess for what they are, we think some of them might be magnetars, which are very highly magnetized neutron stars that tend to explode a lot, and I could talk your ear off about that, but I know we don't have so much time. So I think that's one possible, but I think there might be many types of sources that can produce fast radio bursts.
Actually, I want to ask Simon to maybe just comment on this. Maybe you are also a renowned astrophysicist, and what's your view on this?
What they are?
Yeah.
I'm waiting for Vicky to tell me.
Okay. So unfortunately, I was told that our time is up for this panel discussion. I think it's really too short. I wish we had more time. In fact, I think given the interest in the new research tools, especially AI, I see a lot of interesting discussion, and each one of you seem to have your own views about it, and we can go on forever, but our time unfortunately is up. So let us thank our panelists again for this wonderful discussion. Okay.
Thank you very much to our three laureates, and a big thank you to Professor Wang for chairing the panel for us. Thank you. Thank you, ladies and gentlemen. Please be seated off stage. And for those of you in the audience, this wraps the first trending topic session. As you will notice that there is refreshments outside awaiting us. Please do be back at 4:30 p.m. for the second part of trending topics on AI and the teaching of science. So we'll see you back in roughly 15 minutes. Thank you.